Hi, I am Zaridatul Aini Ibrahim. I am from the Department of Pharmacology from Faculty of Medicine at the Universiti Malaya. My award-winning project entitled Investigating the L1-alpha OPG pathway in promoting autophagic trail resistance in colorectal cancer. Now let me tell you about the statistic of colorectal cancer. Now let me tell you about the statistics on colorectal cancer. GlobalCan 2018 have reported that colorectal cancer is the third most commonly diagnosed cancer in worldwide, and it is the second highest cause of death in cancer patients. And in Malaysia, there are around more than 6,000 of newly diagnosed cases associated with colorectal cancer, and approximately more than 460,000 deaths in cancer patients was related to colorectal cancer. Now these figures are very alarming, and we have to know that the 5-year survival prognosis rate of colorectal cancer is highly dependent on the staging of the disease. As you can see here, while the survival rate of colorectal cancer patient at stage 1 is high as 90%, it can drop dramatically to about 10% in cancer patient that is associated with distant metastasis or late stages of cancer. So this tells us that advanced colorectal cancer remains a major challenge to treat by oncologists, and this calls for desperate measures to find new effective treatment for the colorectal cancer patients. As we all know, chemotherapy and in combination with surgery are the widely used treatment to treat most cancers, including colorectal cancer. However, not all the time these treatments are effective, especially in patients associated with advanced stages in which cancer has spread around the body. And also, this treatment is associated with many side effects. And in recent years, as more research has been done, cancer treatment has entered a paradigm shift from broad-spectrum chemotherapeutic agents and radiation into a more targeted treatment that can precisely target and kill cancer cells. Okay, one of the more targeted therapy that is currently undergoing clinical trial all over the world for various cancers is called trail-based therapy. So what is trail-based therapy? Trail-based therapy, it acts through the death receptor expressed on the cancer cells to induce cell death. So these receptors are highly expressed in cancer cells, but in normal and healthy cells, they are very lowly expressed or negligible expression. So that is why trail therapy is more selective in killing cancer cells without harming the healthy or normal cells. And currently, as I mentioned, this trail therapy is undergoing clinical trial in many places all around the world. And the example are trail activators and trail antibodies. However, although this sounds very promising, trail therapies have several drawbacks, and some of the mechanisms that have been shown to cause trail resistance in colorectal cancer patients and many other cancers are increase in decoy receptors, in which cancer cells express these decoy receptors to mop up the trail ligands from binding to the actual receptors, and therefore they can accept cancer. Sorry, boleh start balik ke? Okay. Okay, sekejap. However, although this sounds very promising, trail therapy have several drawbacks, in which not all cancer cells are resistant, can respond to this therapy. So one of the mechanisms in which cancer cells express or increase the expression of decoy receptors, in which this decoy receptor mop up the trail ligand from binding to the actual death receptor itself, and escape cell death. The other mechanism is through autophagy, in which the cancer cell employ the cell survival mechanism, and they can also escape cell death from this mechanism. Research has shown that there is a link between trail resistance, autophagy, and damage-associated molecular patterns molecule. So what are DAMs? In particular, our group is interested in studying about this molecule, and our group in particular are looking at this protein, interleukin-1 alpha. So what are DAMs? They are secreted in response to cellular stress, infection, and inflammation, and they can initiate and perpetuate inflammatory responses. And in cancer, DAMs have been shown to trigger and promote the development and progression of cancer. Interestingly, our group have shown that interleukin-1 alpha can be secreted from the colorectal cancer cells. And what we want to do in this project is we want to investigate how interleukin-1 alpha could promote trail resistance via the two mechanisms that I mentioned just now, through increase in decoy receptors that can mop up the ligand and cancer cell can escape cell death, or through autophagy, a mechanism in which cancer cells employ to survive from death. And how are we going to do this? So these are our preliminary data, and we showed that R1 alpha are secreted from colorectal cancer cells. And also R1 alpha increase the production of decoy receptors, the one that can mop out the trail ligands. And this is what we plan for the study. So we want to monitor the autophagic activities of the cell, and also we want to look at the cell viability of colorectal cancer cells in response to R1 alpha and trail therapy. And after that, we want to determine whether neutralization of R1 alpha or inhibition of R1 alpha and the decoy receptor would resensitize the colorectal cancer cell to trail therapy, and this could offer new avenues for the treatment against colorectal cancer. Thank you.